Students, today we study plant embryology. In plant embryology, we study today a short note on anther and pollen culture. Anther is a part of stamen containing pollens. Pollen, a fertilizing powder discharged from flowers. Anther. First pics showing the flower, and second pic we see anther. History of pollen and anther culture. W. Tulek first observed that the mature pollen grains of ginkgo biloba can be induced to proliferate in culture to form haploid callus. In India, two scientists, Guha and Maheshwari, first reported to direct development of embryos from microspores of Datura inoxia by the culture of excised anther. Morgan and Nietzsche obtained complete haploid plantlets from anther culture of Nicotiana tabacum. Anther culture, culturing of anther obtained from unopened flower bud in the nutrient medium under aseptic conditions. Callus tissue or embryos from anther that gave rise to haploid plantlets either through organogenesis or embryogenesis. Pollen culture is another process. In pollen culture, we take it direct pollens in vitro technique by which the pollen grains, preferably at the ununinucleated stage, are squeezed out aseptically from the intact anther and then cultured on nutrient medium. The microspores develop into haploid embryos or callus tissue that gives rise to the haploid plantlets by embryogenesis or organogenesis. Culturing of anther obtained from unopened flower bud in the nutrient medium under aseptic conditions known as anther culture. This condition or embryos that give rise to a haploid plantlets either through organogenesis or embryogenesis. Pollen or microspore culture is an in vitro technique in which pollen grains in uninucleate stage directly cultured on nutrient medium. The process of pollen or embryo culture, also known as androgenesis. Androgenesis is in the in vitro development of haploid plants originating from totipotent pollen grains. Totipotent pollen grains means pollen grains have a capacity to regenerate complete plant from a pollen grain through a series of cell division and differentiation. Androgenesis is basically two types: direct androgenesis, indirect androgenesis. First, direct androgenesis. The microspores behave like a zygote and undergoes a chance to form embryoid which ultimately give rise to a plantlet. Indirect embryogenesis. The microspore divide repeatedly to form a callus-like tissue which differentiate into haploid plantlets. Principles for anther and pollen culture. The production of haploid plants is to exploit the totipotency of microspore. Totipotency is a term which defines to a cell's capacity which have to regenerate a complete plant from a cell. This term is known as totipotency. In this process, the normal development and function of the pollen cell to become a male gamete is stopped and is diverted forcibly to new metabolic pathway for vegetative cell division. Various factors influencing anther culture. The first one is genotype of donor plant. The genotype of the donor plant plays a very significant role in determining the frequency of pollen production. Second is anther wall factor. The anther wall provides the nourishment in the development of isolated pollen of a number of species. Second factor is culture medium. Third factor, sorry. For anther culture, medium requirements vary with genotype and of the anther, as well as condition under which donor plants are grown. Incorporation of activated charcoal into the medium has stimulated the induction of androgenesis process. The iron is in the medium plays a very important role for the induction of haploids. Potato extracts, coconut milk, and growth regulators like auxin and cytokinin are also used for anther and pollen culture. Other factors like stage of microspores, anthers are most productive when cultured at the uninucleate microsporic stage. For example, barley, wheat, rice. Anther of some species give the best response if pollen is cultured at first mitosis or later stage. For example, datura and tobacco. Effect of temperature. Temperature enhance the induction frequency of microspore androgenesis. The low temperature treatment to anther of flower bud enhance the haploid formation. The low temperature affects the number of factors such as dissolution of microtubules, lowering of abscisic acid, maintenance of higher ratio of viable pollen capable of embryogenesis. Physiological status of donor plant. Physiological status of donor plants such as water stress, nitrogen requirement and age of donor plant highly affect the pollen embryogenesis. Plants which starved of nitrogen may give more responsive anthers compared to those that are well fed with nitrogenous fertilizers. In this week diagram, we saw the process of anther and pollen culture. Procedure, we first study procedure for anther culture. First we collect of an unopened flower bud. Then we surface sterilize between 80 and HgCl2 mercury chloride. Anthers exist from flower buds and kept separately. Then anthers in first meiotic division is selected by acetocarmine test. Inoculated in the medium containing glutamine, alserine and inositol. Incubated the culture at 25 degrees Celsius for around 15 days. Here anthers grow into embryoids. Embryoids transferred to rooting medium under 3000 lux illumination after 4 to 5 weeks. The embryoids become plantlets. After acclimatization is the process in which plantlets are adapted to environmental conditions. After acclimatization process, the plantlets or embryo are transferred to greenhouse. Procedure for pollen culture. Anthers collected from flower buds and pollen grains are isolated and inoculated in the nutrient mediums with the concentration of pollen 0.5 ml. Niche medium is the best suitable for pollen culture. Anthers are placed on the medium. Sometimes nurse culture may be used. A paper dish is placed over anther or callus. 0.5 ml pollen suspension is poured on the dish. Petri dish covered with lid and incubated at 15 degrees Celsius for around 4 weeks. Pollen grains grown into individual clones with around 60% efficacy. Clones are transferred to callus induction medium to produce calli. Calli transferred to suiting medium and then to rooting medium. Suiting medium and rooting medium both are differentiable according to requirement to produce plantlets. Advantage of pollen culture over anther culture. During anther culture, there is always the possibility that somatic cells of the anther that are diploid will also respond to the culture condition and so produce unwanted diploid caloi or plantlets. Sometimes the development of microspores inside the anther may be interrupted due to growth inhibiting substance leaking out of the anther wall in contact with nutrient medium. Importance of pollen and anther culture. Utility of anther and pollen culture is for very important for basic research and cytogenetic studies. Study of genetic recombination in higher plants. Study of mode of differentiation from single cell to whole organism, how this is happened. This we can study through pollen and anther culture. Study of factors controlling pollen embryogenesis in higher plants. Formation of double haploid that are homozygous and fertile. This is very important utilization of pollen and anther culture. For mutation purpose studies, for plant breeding and crop improvement programs, anther and pollen culture are very important. To obtain the secondary metabolite like products, for example, hyoscyamus niger obtained by anther culture having higher alkaloid content. Haploids are used in molecular biology and genetic engineering experiments. Example, haploid tissue of Arabidopsis and Lycopersicon have been used for the transfer and expression of three genes from E. coli. So these are the some importance of study of anther and pollen culture techniques. Thanks.